Hello guys, welcome to Statistics Bio 7. I'm Dr. Mohanathanari. The channel fully focused on the biostatistics data analysis and data visualization related on video post on the channel. If you like the channel video, click to the subscription button. Support to me, join to our channel membership. Let's go to the video section. Now we are the discussion topic for autocorrelation in past statistical software. So what is the autocorrelations? The autocorrelation is a statistical measure that assesses the degree of similarity between a given the time series and a lagged versions of itself over successive time intervals. In similar terms, it quantifies how current value in a dataset are related to past values. If a time series is strongly autocorrelated, it means that the past value have a significant influence on future values. So the definition of autocorrelations, the correlations of signal with a delayed copy of itself as a functions of delayed lag. The time interval by which the series is shifted. So the autocorrelation functions ACF, a plot of autocorrelation coefficients as a functions of the lag. Usage in biological sense of autocorrelations. The autocorrelations has several applications in biological science, particularly in the study of temporal patterns and spatial distributions. Here are some key usages in biological science of autocorrelations. The first one for the population dynamics. The monitoring trends autocorrelations is used to study population trends over the time, helping to identify cycling the patterns, growth rates, and the effect of environmental changes. Predictive modeling by the understanding past population trends. Scientists can develop predictive models to forecast future population size and dynamics. So the next on ecology, the species distributions, analyzing the spatial distributions of the species within an ecosystem to understand clustering, dispersions, or random distributions patterns. The temporal analysis, assessing the seasonal or annual cycles in species behavior such as migration patterns, breeding seasons, and resource utilization. The next one for the genetic and genomics, gene expressions, studying the temporal patterns of the gene expressions to identify regulatory mechanisms and rhythmic patterns. The genetic linkage analysis, the understanding the inheritance patterns of the genetic thread over generations by examining the autocorrelation of genetic markers. Epidemiology studies, the disease outbreaks, the tracking the separate of disease over time to identify the periodic outbreaks and seasonal trends. The public health monitoring, evaluating the impact of intervention and control measure on the incidence of disease. Neuroscience, neural activity, analyzing the firing patterns of nerves over the time to understand rhythmic brain activity and synchronization. The behavioral studies, examining the autocorrelations in animal behaviors to study habitations, learning, and repetitive actions. The examples of the analysis, the genetic and genomic, the genetic linkage data analysis. So the autocorrelation in genetic and genetic linkage data analysis in past statistical software. The genetic linkage analysis is a method used to study in inheritance patterns of genetic traits over the generations. It involves examines the correlation of genetic markers to determine whether certain alkyls at different loci are inherited together more often than would be expected by change. Autocorrelation analysis can be applied to genetic data to understand the degree of linkage, disequilibrium between the genetic marker. So the left side of the example data set in the table format for genetic linkage analysis. So the example data, let's generated a simple example data set for genetic linkage analysis. Assume we are studying the inheritance patterns of the genetic trait across the 10 generations. So the generation 0 to 10. We have the data 10 genetic markers. So the genetic marker 1, 2 and 10. And we want to analyze the autocorrelation of the marker to determine the linkage. The data set used for the PAS statistical software. So open the PAS statistical software. The data already entered in the past statistical software. So now we are the analyzed for the genetic and genomic genetic linkage analysis data, the only the analyzed in the marker 1. So the select for the marker 1 column, go to the time series menu and go to the correlations and select the auto correlations. So now the open the new tab for the auto correlations in PAS statistical software. So this is for the plant of the autocorrelation in PAS statistical software. The autocorrelation, the plant type available in the 3 types. So the first one for the standard in the line chart, next one for the bar graph, and the third one for the point chart. So the standard selected for the line chart. So then next additional options for the 95% of confidence lines. So this is for the green line for the 95% of confidence line. So the next one options for the tab of the number tab in the auto correlations. This is for the table format in explanation the value of the plant. So this is XHS for the lag options, the starting value 0 to end of the value 4. So the number of the tab, the lag value starting the 0 and 4 end, and the blue line is a correlation value and the green line is a confidence value. So this is green line and the blue line. So this is for the auto correlations analysis in PAS statistical software. Okay, if you continue section of the interpretations video sections, I will upload the next one video. The video interpretation video link mentioned to the description box. If you any doubt on this video, post on the comments in the comment box. If you like this video, click to the subscription button and support to me, join to our channel membership. Thank you for watching this video.